Hello, everyone. Welcome back to my video tutorials for single-cell RNA sequencing data analysis. Today, I'm going to show you how to use the double detection package to identify doublets. So, identify doublets is an important step to perform quality control in our analysis. If you don't remove doublets in your single-cell RNA sequencing data, it will generate separate cell clusters in your UMAP, which will affect your downstream analysis. So first, let's load all the packages for today's analysis. If you haven't installed the doublet detection package, you can use pip install doublet detection. Now we can load the demonstration data. I'm going to use the PBMC 10K cells dataset from 10 times genomics. You can download the data using this link if you want to use the same dataset to practice, or you can use your own dataset. So I downloaded the data already. We can just use the read 10 times H5 function to load the data as A data. At the same time, we make sure the gene names are unique. Then we can have a look at the A data. You can see we loaded the data as A data. At the moment, we have 11,769 cells and 33,538 genes. In the A data, we have the variable names, gene IDs, feature types, and the genome. So normally we perform 3 steps quality control. First, we can filter out the cells that have detected gene lower than 100, and also we can filter out genes that are only expressed in 1 or 2 cells. So let's filter the data first. You can see after filtration, we only have 11,643 cells now, and we only have 20,293 genes. So in next step of quality control, we can analyze the quality control matrix, then subset the data. First, we need to get the gene information for mitochondria genes, ribosome genes, and hemoglobin genes. Let's get the gene information. Then we can calculate the quality control matrix. After that, we can use the volume plot to look at the quality control matrix. For example, here we are going to look at the N genes by counts, total counts, and the percentage mitochondria DNA. Let's make the volume plot. You can see we generated 3 volume plots. The first one is the volume plot for N genes by counts. The second one is the total counts, and the third one is the percentage of mitochondria DNA. So normally in this step, we will subset the data, just keep the cells that are in the volume shape region. Because today I'm going to demonstrate the doublet find function. Normally the doublet has high N genes by count and also high total count. So we are not going to subset the data now. We will go to the third step of quality control, doublet find, without subsetting the data by quality control matrix. In this way, we can more clearly see the doublet in the dataset. So first, we need to set the boost classifier for doublet detection. You can see we can set the n iterations as 10, clustering algorithm as no 1, and we have other parameters here. Then we will run double-need detection using the double-need detection Python package. So we will get the data for double-need and the double-need score. So now we can run the double-need detection. Okay, it is done. You can see it takes 1 minute and 15 seconds to run the double-need detection. So next we can look the data. First we can plot the convergence to see the double-need numbers according to different threshold. Let's make the plot. Okay, you can see here is the number of iterations against the number of predicted double-needs. You can see the iteration 1 has the highest double-need. It is over 800 double-need in this dataset. So next, we can look at the number of predicted double-need at different threshold combinations. This is a heat map. Let's run the code to generate the heat map. Now you can see we have the heat map. The Y labeling is voting threshold. We set it at 0.5. But here you can see from 0.3 to 0.9. And the x-axis is the log 10 p-value. So you can see from the legend the predicted double needs. If the log p-value is null and the voting threshold is null, then we have a high number of double need showing in yellow color. If we set the p-value is very high and the high voting threshold, then we detect a very low number of predicted double need. So we have the double need and the double need score data. Now we can add those data into the A data observations. We can add the double need as the double need and the double need score as the double need score into the A data observation. Now we can have a look at the A data. You can see here in the observations, we added the double need score. So we finish the double need detection. Next, we can perform a standard scRNA analysis to cluster the cells. Then we can visualize the double need on the UMap. So we're going to run the standard workflow, normalize the data, logarithmize the data, find the highly variable genes, run PCA, compute the nearest neighbor graph, and run UMap. We are going to cluster the cells using the native method. Then at the end, we can generate the UMap. Let's run the analysis. Okay, you can see we finished the analysis. Here is the UMap. In total, we have 19 cell clusters here. Because we have the double need and the double need score information in the observations, so we can look at the cell clusters by the latent cell clusters and also the double need and the double need score. We run this code to generate 3 UMap. You can see here, left-hand side is the latent clusters. Then the middle UMap will show the double need. You can see all the double needs were indicated by yellow color. Single need were labeled as dark blue color. And on the right hand side, you can see the double need score. You can see those cell clusters, they have a high double need score and they are labeled as double need. So from the UMap, you can clearly see 3 cell clusters. They are double need. Those cell clusters could be heterogeneous doublet. Other doublet you can see they mixed very well with single knit. So those doublet could be homogeneous doublet because they express similar genes as the single knit. You can see these three clusters. If you don't remove it, it will be difficult for you to label the cell clusters correctly and also will affect your downstream data analysis. So now we label the doublet, we can remove the doublet from the data. I showed you we have the doublet information in the A data observations. We can have a look at the doublet information. You can see the cells that labeled as 0 are single knit. If the number bigger than 0, they are double knit. So we can subset the data using the metadata. If we set the double knit bigger than 0, we will get all the double knit. We can subset it as B data for demonstration. Let's subset the double knits. You can see here we only have 763 cells when labeled as double knit. We can generate a UMAP for all the double knits. Let's run. You can see here, left hand side are the 763 double knits, and the right hand side are the UMAP for the double knit score. Here I just want to show you we can see the double knits, but in reality we need to get rid of double knit. So next we can subset the data to keep all the single knit for downstream analysis. For the demonstration, I'm going to subset it as C data, but if you want, you can subset it as A data. So now we subset the data to keep all the single knits. Let's run. You can see we have 10,880 single knit in this data set. So after subsetting the data, we kept all the cells as single knit, but the column name is still double knit to show all the single knit in the data set. So we can generate a new metadata column as single knit. Let's run. You can see we added a new column as single knit. If you want, you can also delete the double knit column. Now we can generate a new UMAP to show the single knit. So we can count the cells by single knit and double knit score. Let's generate the UMAP. You can see on the left hand side, it is the UMAP for all the single knit. And on the right hand side is the double knit score. Now you can see we removed all the double knit in the data set. You can use this data set to perform further analysis. Okay, that's the video tutorial for double knit detection using the double knit detection package in Python. I hope my video tutorial could help your data analysis. If you like my video tutorials, please share my videos with your friends. Thank you.